the next speaker, Dr. Richard Novak, who is CEO and co-founder of Unravel Biosciences. He will be presenting on Patients First, Rapid Prototyping with Repurposed Drugs to Accelerate and Clinically De-risk Therapeutics for Leigh syndrome. So it's all you, Dr. Novak.
Thank you. It's a pleasure to be here. Thank you to Casey and the Cure Mito team for inviting me. And these presentations have been wonderful. So wonderful to attend and participate. So we are a company that spun out of Harvard about 3 years ago, and we really put patients first in our drug discovery and development path. We take an engineering approach to therapeutics that I want to describe. And we just recently started working on Leigh syndrome. So just to take a step back, especially for the lay audience, I think the immensity of the scale of the problem that we're dealing with is worth understanding. So let's talk about skyscrapers. I know that's why everybody's here. So the whole idea is that we build skyscrapers all the time. What's really exciting and interesting to me is that to build a skyscraper takes about the same amount of time and money as a drug. Puts everything in perspective, especially for rare diseases. The difference is that even despite these astronomical costs, we are very successful at building skyscrapers. Not so much with drugs. Being very generous here on the statistics, you basically have to build 20 skyscrapers to get one to stand up. That's the equivalent. That's what we essentially are doing in therapeutics. It just doesn't make sense. Well, we see really wonderful opportunities in applying the same engineering approach that is very successful in literally every industry outside of therapeutics, and make products more efficiently. Now, a drug is just like any other product, potentially. And that's really what Unravel Biosciences has set out to do. And why do we take this engineering approach? Why do we move quickly? Well, patients can't wait. We've heard that countless of times just today. Obviously, depends on the gene, depends on a lot of other factors, but we can't wait around for 20 years to try to understand the deep biology. Even though that's very much worth doing, we need help today. We need to move something forward. And so our mission is really to transform how we do drug development and drug discovery by clinically de-risking new targets that we discover using existing drugs. Now you might say, oh, this is repurposing. Well, repurposing can be an intermediate step here. We think that new molecules are going to be the long-term solution, but of course that takes time, takes a lot more resources. But let's do something today. Let's make sure we're on the right path. Just like with engineering, are we moving in the right direction? If not, let's pivot really quickly without spending 10 years to get a new molecule into the clinic. And so what we do is really make sure that all patients, and really I'll talk about the scale of what we do, but all patients have access to safe and effective treatments, and really no matter how rare, complex, or unique the needs are. And we're doing this in a very data and ethics-driven approach. And so bottom line is we build drugs that work. The whole idea is that we don't understand biology, or not nearly getting close to that, though we're moving in the right direction for sure. And yet patients need help today. So what can we do? Well, even if you don't understand something, you can simulate it at times. And again, this has parallels in physics, has parallels in engineering and other approaches. And so we built a tool called BioNav, and that's a computational AI-enabled platform that essentially is a model of human health. It essentially connects, and there's a real picture that we attempted to put together, showing sort of staggering complexity. It's really hundreds of millions of interactions of genes and drugs and really these probabilistic models of health that let us simulate what's happening in a person and what's the response to a drug. And this was developed at Harvard as one of the things that we spun out, funded by a lot of DARPA and other DoD funding. And really what it does, it's an AI-based way to ask, if you have patient data, how can you restore this abnormal network, whatever that may mean for that person? How can you restore that back to health? And then you can get various predictions, and that's what I'm going to focus on in this talk. But what we also do, we make animal models, because for most diseases out there, especially in the rare disease space, there are no animal models that you can really ask the questions you want to, which are clinically relevant questions: seizures, sleep, kind of organ level interactions. And that's what we do. We build in vivo screening models using tadpoles, of all things, frog embryos, and ask those questions. But really what we're focused on, we have a chief medical officer. We're looking at how do we test this in actual patients? Ultimately, that's the problem we're trying to solve, not trying to make an animal model better, though that's a good stepping stone. We want to make patients better. And so what does that look like? Well, how can we do these proof of concept studies to de-risk the novel mechanisms, novel targets that we find, but in actual patients? And that's where existing molecules come from. Those are ones you can give a patient today and ask the clinical questions you want. But ultimately, we are developing new drugs that are now tailor-made for whatever the disease may be. We focus on rare diseases primarily, but from there expanding out to group other rare diseases together by shared mechanisms, but even non-rare diseases. And the whole platform is really founded on this concept that a disease is really a product of the genetics. Yes, the mutated gene, of course, but all the other 25,000 or so genes can't be ignored. And most of them are non-pathogenic, but together with a mutation, they can have very different effects. The environment, this includes medication, microbiome, sleep, all those factors will have some impact on the disease. And then time. Patients at birth are different from patients a year later or different from patients 10 years later. There's accumulated disease burden. There've been therapeutic effects. And so that's why we use RNA as opposed to DNA to drive this platform. RNA actually reflects all of those dynamics, and we can, using that, model human health. And essentially, think of this as a Google Maps for health. You're trying to go from, let's say, your home to work. You don't really care about the differences. You care about the path in between, and that's what RNA will start telling us. And so just to illustrate how this works, we have some conceptual healthy network. Actually, the illustration behind it is real data from Rett syndrome, but the lines here are just illustrative. And there, let's say there's a mutated gene. In this case, it actually is a real gene that's mutated. It has some disruption to this network, very complex. Maybe only a fraction of that has been studied to date, right? We need to fix it. Well, what we look for, we essentially simulate 40,000 molecules and see how do we restore that network back to health. It could be going through that mutated gene to some extent, but more often than not, biology is very plastic. And so we can look at all these nodes and these networks, just like if you're trying to go from home to work and your typical, interstate bridge, interstate has a bridge that has construction and backs up traffic, you might want to go off the highway and find an alternate route. That's what we do, among other things. And so we can rebalance that network, and essentially it's about that function, the function of the network, and how can we restore that without making any assumptions, right, about how that should happen or not. And I won't go into detail on the slide, but basically this approach has really shown very strong promise, and we have validation data for on all these offerings tied to drug development, from discovering a target that could be therapeutic, finding molecules that could actually do what you want to do, looking at safety, right? Looking at safety in a very heterogeneous patient population. We've heard that a number of times today. One Leigh syndrome patient is not like the other. There's over 100 genes involved in Leigh syndrome. You would assume that they're not all the same. But even looking at life cycle management, looking at making these rare diseases commercially viable in a strategic way, and then looking at patient stratification. Ultimately, it's individual patients that respond to drugs, and we need to work with them from the beginning to really make sure that we're developing treatments that can have a positive impact. And to that end, we're really building effective drugs by enabling this patient-centric ecosystem. Just like computationally, we are looking at biology as a system, as a network. That's the same concept applies for drug development, drug discovery. Having a fancy new tool to kind of plug into a drug development pipeline is fine. It can add some value. But ultimately, we're trying to build that skyscraper, right? You can have a better hammer. You still need cement. You still need a better design. You need all these other factors. And so we're looking at it from a systems level perspective, but with patients really driving the whole process. So I already alluded to the platform, right? It's the Google Maps for health, and really simulating these networks of genes in a healthy system, and then using patient data to move that forward. And that's really where patients come in. It's patients and not diagnoses that respond to treatments. And so we partner from the beginning using patient data to drive programs forward. And I'll have several examples in a moment to show. But in keeping with that kind of broad ecosystem that we're building, we, of course, need molecules or some sort of treatments to then translate into the clinic and into the market. That actually is how patients will have access to any of these discoveries. And that's where the pipeline comes in. And so we're actually developing multiple candidates sort of in parallel, trying to drive very rapidly at making impact across multiple rare diseases at once in a very efficient way, because there's about 15,000 rare diseases. We can't really go one at a time. And then the last part is potential. And the idea is that, rare disease is tough, but where there's something that's hard, technically, there's also a lot of opportunity for patients, opportunity for improving how we do everything else. And the idea is that we're identifying previously undrugged targets that could open up new avenues in other diseases, maximizing how we get clinical trial success. from recruiting patients to enrolling responders to minimize side effects and maximize the outcome for patients as much as the sponsors and drug companies, and then expanding drugs into new indications. So this could be for new or existing molecules. Let's help as many people as we can in parallel, as opposed to this kind of serial one-by-one approach. So it's really this new ecosystem that we're building, again, centered on patients. And to that end, we had to develop a new business model. So even though we are a biotech, we essentially developed these two business models. So because families and foundations came to us, we decided to offer essentially a service arm. We're not a service provider, we're not a CRO, but we felt that given that we use existing molecules to ask the critical question, does it work? In actual patients, involving those patients is absolutely essential to the process. And so this is really just a cost reimbursement model. We charge what it costs us to work with families, and essentially most folks are looking for rapid drug repurposing for rare, ultra rare, even undiagnosed patients. And we work all over the world. We work on literally every continent using the process I'll describe in a moment. Now, that initial discovery work can then be sort of strategically translated into the biopharma arm of Unravel, which is your classical biotech, but driven by clinical data, right? The idea is that it's a biopharma approach focused on new drug discovery to address these unmet patient needs where existing molecules may not exactly align for various reasons. Could be side effects, could be toxicities, could be availability, that you name it. We're really kind of straddling both of these areas to optimize how we actually make an impact in the clinic. And so how we work through RareShift, again, it's this computational platform, is Google Maps model, but how do you digitize a patient, right? How do you get a patient into a computer? And that really comes through the nose in our case, the obvious choice. The idea is that we need patient data, patient cells, patient RNA, and given this network approach, much to our surprise, we found that we can work off of really any tissue in the body, especially for a lot of neurologic diseases. You're always working with surrogate samples, right? That you won't really have access to the brain directly. And so we said, okay, what's the easiest to collect? And thanks to COVID, silver lining, people know how to swab. And it's FDA approved for self-collection. We have this kit, and it actually preserves RNA really nicely. We've mailed samples all around the world, or collection kits. We get genetic insights for those patients versus their respective family controls. And we can do this analysis. Now, that patient is part of our clinical research, and we can plug them into this AI-enabled analysis pipeline, understand the condition as it evolves, even effects of treatments, either ones that they're on or ones that they may be considering. Basically understand how patients would respond to given therapeutics. And especially find potential repurposing avenues. That's really the main thrust of a lot of these programs. And so these nasal swabs that I alluded to, really it's a home collection kit. People self-swab, or in the case of pediatric patients, the parent or caregiver swabs, very easy, painless, and it's accessible. So we essentially have a preservation solution in there that was, again, developed by a partner that inactivates pathogens, so it goes through customs really easily. It's not considered clinical samples, and you don't need a cold chain. So all of a sudden that you can just ship through regular FedEx or other shipping services globally and bring in patients from all around the world to participate in either personalized repurposing or sort of population level analysis. And I'll give some examples in a moment. And then the best part is that this actually correlates really nicely with what we would get with other more invasive tissue sources. And so how does this work, right? Here are two projects that we completed recently. One is in MEF2C, so it's a very complex regulated gene, kind of transcription factor regulator. And we found that all these patients had the same diagnosis. They came in with a haploinsufficiency. We found that two of the patients were unlike the three over here, and then the three were actually relatively similar in terms of their drug prediction profile to all the different models. They're all knockout models of some sort. Well, what it turns out later, we found that the point mutation patients were all in this cluster. And at the time, it wasn't clear what the point mutations do. Now it's known that they're actually a gain of function. So it's really not a haploinsufficiency for all 5 patients. The other 3 likely are. But these 3 are actually very different. And so we saw that diversity in the population, not knowing anything about any of the patients. just based on the nasal swabs. And so this actually helps determine the strategy in some of these populations. And then similarly, for AP4, it's actually 4 different genes all tied to a single protein complex. And we showed that the gene or the sex of the patient didn't really matter. What does matter, likely, we're doing another survey to follow up, but likely it's actually the severity of the disease, which is actually not driven by the gene. There are other factors that are involved that are yet to be discovered, and yet we can see them. And then what's really exciting that we can go even look at sort of clinical effects. These are existing drugs, and so families oftentimes choose to actually test them out themselves with their clinicians. Again, we have to be hands off. We cannot diagnose or treat diseases directly, but we can certainly provide data that may be required. There's a family with Rett syndrome that started taking vorinostat that we identified for Rett syndrome using the nasal swab, and the patient is doing significantly better now about 9 months out, continuing to improve. And similarly for a completely different disease, very different molecule, they saw very rapid improvements in about 2 weeks for sleep, ADHD, and actually recovering receptive speech. So this patient was unable to communicate either direction. And this was confirmed by sort of independent blinded observers. And then we can go all the way to clinical trials. This work with Mass General Hospital, finding novel mechanism, but an existing drug. And so we work with over 25 programs. But what's really exciting, I think, for this audience, we started working on Leigh syndrome thanks to the Cure Mito Foundation. So we work on 5 genes. One is actually separate from Cure Mito. And they're just highlighted here, and they really span beautiful diversity of the mitochondrial complexes and even a non-complex protein, ECHS. And we found actually in initial work, we're just now getting some of the patient data, but from some initial public data sets, we've actually confirmed ProBcol efficacy computationally. We also predicted toxicity of 2 molecules that we were considering for another disease. And NDFS4 was used as a mouse model because there was no mouse model. And we predicted actually for it to not work. And sure enough, it did not work. Mice died earlier than the untreated ones. So it's sort of a good negative validation. But we're also predicting those same molecules to actually work for other disorders besides NDFS4. And so what we're doing now is working with 20 patients or more, collecting the nasal swabs, and for the patient and for the healthy family member, generating that data. That will be all shared. So that's data for research purposes. We will use that to find potential treatments and potentially other insights. But then the real key here, especially in this very heterogeneous patient population, is to make sense of Leigh syndrome to some extent. Which patients are like others and which ones are not. How many categories are there really? We're finding from NDFS4 that they really the complex even doesn't necessarily drive those responses. Just like we saw the genes in the AP4 complex does not necessarily drive the gene response or the drug response. And so in conclusion here, we developed a patient-driven therapeutics prototyping path. So RareShift is really the data generation prototyping, asking that critical question, does it work in an actual patient? And of course, the steps to get there. And then we as Unravel are developing novel therapeutics for the targets that we discover. So thank you for the opportunity again, and I look forward to any questions.
Great. We'll take questions at the end after all the presentations. Thank you.